So, good afternoon, everyone. I'm Rishabh, and today we'll be starting with the discussion of the Introduction to the Cell Biology NPTEL course by Professor Nagaraj Balasubramanian and Professor Girish from IISER Pune. So, I'll be discussing about various aspects that will be studied in different weeks or different modules of this particular course, and I have a different teaching pattern. So, I'll be teaching you in terms of various question and answers, and I prefer to be more interactive so that I would also know that whether you guys are understanding or not. So it would be good if you guys interact during the question answer itself, and I'll be happy if you guys can actually ask whether you have understood or not, so that I'll be able to answer your questions at that particular time only. So we'll be starting with the first question, that is, the picture below depicts the flow of information in cells. The central dogma, which pathway showed in the red numerical depicts the stage that represents the work of RNA-dependent DNA polymerase? So here is the pathway shown. So what can be the correct answer? Okay, some said 7, some said 4. Okay, so the correct answer is option 4. That is the fourth option. And now we'll be exploring. So first of all, what is a term dogma means? Okay, so the term dogma basically means any hypothesis that doesn't have any experimental evidence. Okay, so it was given by Sir Francis Crick. Okay, and he suggested that this is a hypothesis based on its understanding at that particular time that the DNA, that is the major storage molecule for genetic information, can actually transmit its information in the form of an RNA, that is also a nucleic acid, and the process by which it transfers it is called as transcription. Now, this RNA to express in terms of a protein, it's called as translation to show the phenotypic effect. Okay, so basically, this central dogma explains that DNA, that means your gene, how it is showing the phenotypic effect in the body. Okay, so let's see on the different terms that are giving in the particular diagram. So when we see the DNA, it actually replicates itself. That is one. Then it can transcribe itself from DNA to RNA. Okay, so that means it is actually, this seventh step is transcription, that will involve DNA dependent RNA polymerase. Okay, now when RNA is found, then RNA actually gives rise to RNA, that means it's RNA replication. Okay, and when this RNA can be converted back to DNA, so this is actually done by the enzyme called RNA dependent DNA polymerase. Okay, so this is actually reverse transcription. Okay, and this term was given by Temin. That is why it is called as Teminism. Okay. Now the final step, when the RNA changes to protein, this ninth step is called as translation. Okay, and there are hypotheses that this protein can also change back to RNA. This protein can also change back to DNA, and DNA can also produce proteins directly. Okay, but these are hypotheses, and there is no experimental evidence yet for these particular pathways. So this is the basic. So this is the basic central dogma of the molecular biology that is explained here. Okay. Okay, so let's move on to the next question, that is, the picture below depicts the flow of information in cells, that is the central dogma, which pathway shown in red numerical has not yet discovered in eukaryotic cells. So this is the same pathway that have been shown in the last question. So which of the following pathways is not discovered in eukaryotic cells yet out of the given options? Someone said second, then any more answers? Okay, seven, then 4, 7, 2, 6, then okay. So let's discuss. So as we see that DNA, the major genetic storage molecule, converts its information to the RNA. That is taken care by the process called as 7. Okay. So this process actually occurs in both prokaryotes and eukaryotic cells. Okay. So this seventh one will occur in every organism. Now, this the conversion of DNA to DNA, that is replication, also occurs in both prokaryotic and eukaryotic cells, right? Then, following this, if we go from RNA to protein, so this information is also being converted in both prokaryotic and eukaryotic cells. So this is the general information pathway that happens in every organism. Now, talking about the RNA conversion to DNA, that is called as reverse transcription. That also happens in like both eukaryotes and prokaryotes. Okay. Now, where does it happen in eukaryotes? It happens in the telomerase activity. Okay. So it happens in eukaryotes also. So it is not an exception. So if we see the options, So the seventh option is DNA dependent RNA polymerase thing that happens in eukaryotes. Then the fourth one also happens in eukaryotes. So this option is also false. Now coming on to the replication of RNA to RNA. Okay, so this doesn't happen in eukaryotes primarily. Okay, now moving on to the conversion of protein to RNA. This also doesn't happen in eukaryotes. Conversion of protein to protein. This also doesn't happen in eukaryotes. And the conversion of DNA to protein and vice versa, from protein to DNA. These pathways are also not yet discovered in any organism. Okay, so the correct option would be option 2 and option 6. Okay, so here option 3 is written, that is option 6, and option 2 would also be mentioned here. So it would be option 3 and option 2 that are not present in the eukaryotic cells. Okay, so is the explanation clear? Any doubts in this question? Okay, okay. So let's move on to the next question. The central dogma of molecular biology describes the flow of genetic information in cells. Which of the following correctly represents the process? So, what can be the correct answer? Okay. Any more answers? So, yes, the correct answer is option number 3, and the explanation I have given just before, that the flow of information takes place from the DNA, the major storage molecule, then it flows from DNA to RNA, then from RNA to proteins via translation. Okay, so this is called as the expression of information from gene to protein. So the correct answer would be option number 3. Okay, so let's move on to the next question. The first accurate model of DNA was proposed by which of the following options are correct? Okay, 1 and 2. Okay, any more answers? So, yes, the correct answer is option number 2. So, the accurate model of DNA was proposed by Watson and Crick. Okay, that is option number 2. Now, what they did was they actually had the idea in mind. Okay, so they actually like they accumulated all the information that was available for the DNA structure. That is the X-ray crystallographic structure of DNA. That was the observation given by Rosalind Franklin and the Morris Wilkins. And then they also taken the data for the Chargaff, and secondly with Polly. So they actually compiled all the data from these information, and they did the double helical structure of DNA. So they actually gave the first accurate model for DNA after compiling all the data from these informations. Okay, and this was majorly accomplished, or it was possible only by the X-ray crystallographic structure of DNA that was given by the Rosalind Franklin and Morris Wilkins. Okay, so it would be correct if these two, first and second, would be the correct answer. Although these two compiled the data and they gave the structure, but it would not be possible with the X-ray crystallographic structure of Rosalind Franklin. Okay, so the correct answer would be option 2 for now. But if like any literature or came or anything will happen, then we have to consider all four for giving the accurate model for the DNA. Okay, so any doubts in this question? So here in the figure, they are actually discussing about the double stranded structure of the DNA, and here, as you can see, these are the nitrogen bases. Okay, these are the nitrogen bases, and here they are connected by the hydrogen bonds. And this person on the left, it's Watson, and the one standing is Crick. Okay, so let's move on to the next question. What does the X-ray diffraction pattern of DNA produced by the Rosalind Franklin suggested to Watson and Crick? So, what can be the correct answer? Okay, any more answers? Okay, then okay, any more answers? Okay, just think what can a X-ray diffraction technique can give to Watson and Crick. Okay, any more answers? Okay, so let's discuss. So the correct answer to the question is option number 2. So the X-ray diffraction pattern actually gave the idea that the DNA has a double helical structure. So this was the diffraction pattern that was observed by Rosalind Franklin and Wilkie. Okay, so what actually they observed was the DNA has a diffraction pattern of like this. Okay, so this is a diffraction pattern that was observed like this. There was a hole in between, and there were layers in the form of an X. Okay, so this actually depicts that there is a double helical structure. Okay, and this angle between these two helix, that is theta, that is actually given by the angle between these two. Okay, and these layer lines that you can see here, that 10, 5, that actually depicts these layer lines. Okay, so this X type of structure that was visible from the X-ray diffraction pattern get us the information that the DNA is a double helical structure. Okay, so this is how they predicted that the DNA is a double helical structure and it has two strands basically. Okay, so any doubts in this question? Okay, okay, so let's move on to the next question. Chargaff rule for DNA states that, so which of the following statements are correct? Okay, option 2, option 2 and 3. Okay, okay, then okay. Any more answers? So most people are centered at 2 and 3. Okay, so the correct answer is option number, sorry, yeah, the correct answer is option number 3. And why it's 3 is that Chargaff rule mainly describes the number of purine and number of pyrimidine that is present in a complementary DNA. Okay, so it mainly talks about the complementarity of the DNA and how it links with it. Okay, so now the wait a minute. So it basically depicts the amount of purine and the pyrimidine that is present in a DNA. Okay, so as you know that DNA has a complementary structure. So on what does this complementarity mean? That once the DNA runs in 5 prime to 3 prime direction, the other direction that would have the 3 prime to 5 prime direction would have the bases that are complementing to the first strand. Okay, so as you know there will be a, if there is a, then on the opposite side it would be T, and if there is G, then on opposite side it would be C. A and T are joined by the double hydrogen bonds, and G and C are interacting with the three hydrogen bonds. Okay. Now, if there are number of A's that are present in a sequence, will be number of T, will be equal to the number of T because they are complementing each other. Similarly, if there are number of G's, then that would also be uh, uh, equal to the number of C's that is complementing each other. Okay, so if we count this, so purines, that is A and G, and pyrimidines, that is T and C, are equal. Okay, because A will complement with D and G will complement with C. Okay, so that is why they are saying that the number of purines are actually complementing, sorry, So the number of purines are actually equal to the number of pyrimidines, or they are in the ratio of 1 is to 1. Okay, and if we see the number of A plus T, that would not be equal to the amount of G plus C, and that will vary between the different organisms. But they will be same in different tissues of the same organism. Okay, so this is the basic Chargaff rule, that just states the number of purines and the pyrimidines that are present in a single double stranded DNA molecule. Okay, so any doubts in this question? The number of hydrogen bonds that are present between the purine and the pyrimidine was not given by Chargaff. That was actually given by Pauly based on the chemical nature of the bases. Okay, so that's why it is not included in the Chargaff's rule. So any doubts in this question? Okay, if not, then we'll move on to the next question. So the two strands of DNA are joined by which of the following bonds? Or what can be the correct answer? Okay, then hmm, okay. So yes, the correct answer is option 3. And as we can see here, the both the strands, okay, here these are the bases. These colored ones are the bases, nitrogenous bases. So they are connected with each other with the help of the hydrogen bonds. As you can see here, this is adenine, this is thymine. So they are connected by two hydrogen bonds. And this is guanine and this is cytosine. These are connected by three hydrogen bonds. So these both are connected with connecting both the strands, and those are taken care by the hydrogen bonds. So the correct answer is option number 3. Okay, so any doubts? Okay, so let's move on to the next question. A nucleotide is composed of. Okay, okay, so the correct answer is option 3. So as you can see here, there are three components of a nucleotide. First is the sugar that is connected with the phosphate group. Okay, so the sugar connected with the phosphate group, and then the nitrogenous bases. Okay, so these three are called as a, when they are combined to each other, are called as a nucleotide. Okay, and what is a nucleoside, by the way? So what will we call it as a nucleoside? Yes. So when we have a base that is connected with the pentose sugar, so that is called as a nucleoside. Okay. So any doubts in this question? Okay. So let's move on to the next question. A linkage between the nitrogenous base and a sugar molecule of a nucleotide is called. Okay. Any more answers? Okay. Any more answers? Okay. Okay. So the correct answer is option 3. And here is the explanation. Okay. So a few things based on the answers that you have given. First of all, if there is a sugar moiety, okay, so this I'm talking about the biochemistry terms. Now, if there is any sugar moiety that is actually forming bond with the other molecule, being it a sugar molecule, a lipid molecule, or a protein molecule, any time that is connecting a sugar moiety with any other molecule, the bond that is formed between these two will be a glycosidic bond. Okay, so this glycosidic bond is actually formed when a sugar moiety is connected to any other molecule. Okay, is it fine? Now, the phosphodiester bond is actually being formed between the phosphate group of one nucleotide and the sugar molecule of the other nucleotide. So that is a phosphoester bond, and because one sugar moiety actually connects two phosphate molecule, so that is why this bond is called as a phosphodiester bond. Okay, this actually connects two nucleotide molecules. So phosphodiester bond is actually helping two nucleotides to be connected to each other. Okay, so now first option is wrong because it connects two nucleotide molecules. Here we are trying to explain what is the connection between the nitrogenous base and a sugar molecule. Now, if we see here, for example, here we are in the first nucleotide, the adenine is connected to a sugar molecule that is a pentose sugar. Now the N of the adenine, because it's a nitrogenous base, the N of the adenine is connected to the C1 carbon of the pentose sugar. Okay, so this, because here a nitrogen is taking place in forming the glycosidic bond, so this bond will be named as N-linked glycosidic bond. Okay, if now, so the correct answer here would be N-linked glycosidic bond. Now I'm giving you an additional information regarding the O-linked glycosidic linkage. Okay. Now what happens in some of the proteins where there is serine or threonine? Okay, that serine has a OH group, right? So whenever a sugar molecule is connected to this OH molecule, then that bond would be named as O-linked glycosidic bond. Why this is O-linked? Because the bond that is formed between the sugar moiety and this OH group is involving oxygen. That is why it is called as a O-linked glycosidic linkage. And here, because the N is involved in formation of the glycosidic bond, that is why it is called as the N-linked glycosidic linkage. Okay, is the explanation fine? And I hope I have cleared the confusion also between the phosphodiester bond and any other linkage. Okay, so let's move on to the next question. Which of the following DNA form is left-handed? Okay. Okay. So the correct answer is option number three. Okay, and here is the explanation. Okay, first I'll explain this question, then I'll go back to the last question. Okay, so uh, this Z form of DNA is the left-handed form of DNA. Okay, and what happens is, so starting from, there are three forms of DNA: the B form, A form, and Z form. B form of the DNA is the most common form that is present. Okay, so both the double helical structure of the DNA also describes the B form of the DNA. Okay, and it is a right-handed structure. And then there is another form, that is A form, that is also a right-handed structure. And there is another form, that is Z form or the Z form, that is a left-handed structure. So here the left-handed one is the Z form. Okay, and here is the major helical pattern of these three form of DNA. So as you can see here, the A form, it's actually right-handed. So the curvature is right-handed in nature. Same is the case with the A form. Okay, although the helicrise base tilt is different in both the cases, because here it's more tilted and it's more constricted. Okay, and here it's more stretchable as compared to the A form. But here, as you can see, the helical structure is left-handed. It's towards the left. Okay, so that's why it's a different form of DNA that has been seen in different organisms. Okay, so is this question clear? Just 1 minute. Which one? Sorry. So the next question is, what role do the major and minor groups of DNA play in the function of the molecule? They allow DNA to be compacted into chromatin. They prevent the DNA strands from unwinding. They are involved in the process of DNA replication, or they enable specific proteins to bind to the DNA sequence. So, what can be the correct answer? So, the correct answer is option number four. That means they enable the specific proteins to bind to the DNA sequence. And here is the explanation. Okay, so here is the major groove of the DNA. As you can see here, it is wider, uh, deeper in nature. Okay, and larger. And this one is the smaller one. That is why it is called as a major groove. Okay, and this one is a smaller and shallower. That is why it is called as a minor. Okay, now what happens is, this major group plays a major role in binding of proteins that are involved in transcription, translation, and replication. Okay, while this minor groups helps in binding of proteins that are involved in regulation. Okay, so this is the major difference between both the groups. Okay, and there are different base pairing patterns and the width of the groups that are present here. So that is different in both major and minor groups. And then the why these groups are actually present. So, for example, if you consider this as a DNA strand, and with as we are talking about replication, so I'll tell you about the replication in brief. So what happens when this DNA has to be unbound? There are proteins that needs to be sitting on on this particular DNA strand so that it can actually denature and the hydrogen bond between the complementary base pairs can actually be broken. So for that, this protein actually sits on the grooves, that is the major groove, and it opens up the DNA. Okay, so these grooves basically helps the proteins to basically sit on the DNA and do their function. Okay, so that is the major function of these particular grooves. Okay. So the next question states that how does the process of DNA replication ensure high fidelity in copying the genetic information? Oh, sorry, in copying the genetic material. So what can be the correct answer? Okay. Any more answer? So fidelity means that anything that the DNA polymerase is actually being doing is doing correctly with minimum errors. Okay. Okay, so the correct answer is option. Yeah. So the correct answer is option 2. That means the DNA polymerase has a proofreading activity that ensures that it correct errors during the replication itself. Okay. The helicase is only is helpful in only the unwinding of the DNA. That means only breaking of the hydrogen bonds. Okay, so it will not provide any fidelity towards the replication process. Okay, so the correct answer is option number 2. So is it fine? The explanation. Okay. Okay, so let's move on to the next question. So what is the significance of the 3 prime and the 5 prime ends of a DNA strand in the process of DNA replication? So what can be the correct answer? Okay. Okay. So any more answers? Okay. So The correct answer is option number 2. Okay, so huh. So what happens actually is, so if we consider the DNA strand, that is 5 prime to 3 prime. So now I want you guys to focus on the chemical structure of the nucleic acid. Okay, so I'll be drawing that now. This is the sugar moiety. Sorry for the drawing, it's not that good. Okay, so this is a base. Here we have the phosphate group attached. Okay, so this is the first carbon, second carbon, third, fourth, and the fifth one. Okay, where there is a phosphate group attached, this is the fifth carbon. Okay, now this is the 5 prime end of the sugar moiety. This is the 3 prime end. Whenever DNA replication happens, it happens from 5 prime to 3 prime, right? So that means this nucleotide has a free 3 prime OH, that is actually being ready for the next nucleotide to bind to it. Okay, so here comes the next nucleotide that also have a base. This is the 5 prime OH. Now this 5 prime phosphate group will attach to this 3 prime group, and from here the 5 prime to 3 prime direction will continue. Okay, so that means the 5 prime to 3 prime direction will continue as such. Now in this case what will happen? Now this nucleotide is joined to the other one through this 5 prime to 3 prime phosphodiester bond. Okay, now this 3 prime of this particular nucleotide is free, and it will help in binding to the next nucleotide. Likewise, the replication happens. Okay, so the DNA polymerase actually adds up the nucleotide in the 3 prime empty group of this particular nucleotide. Okay, that's why it is called as 5 prime to 3 prime direction of the DNA replication. So the correct answer would be option number 2, because the new nucleotide that is added up to the first nucleotide is at the 3 prime position. And if we see to the end nucleotide here, the 3 prime position would be empty. That is why it is called as the 3 prime end of the DNA. Okay. And the third option, that is the 3 prime and the 5 prime ends of the DNA strands, determine the sequence of the amino acids in protein. So this is false. Okay, never the DNA strand actually determine the protein sequence. Okay, so option number 2 is correct. So any doubts? Is the explanation fine for the DNA replication and why it happens from 5 prime to 3 prime direction? Okay, so let's move on to the next question. DNA replication is initiated by which of the following enzymes? Okay, okay. Yes. So the correct answer is option 4, that is helicase. So here is a representation of a DNA molecule. So as you can see here, these 6 subunit, that is a ring-like structure, is of helicase. So what it is doing is, it is breaking the hydrogen bonds that is shown in this green color for the further DNA. Okay, and here it is started to separating the DNA, and here the nitrogenous bases are separated, and these are bound by the single stranded binding proteins, DNA binding proteins. Okay, so that's why the first protein that binds to the DNA that helps in starting the DNA replication is the DNA helicase, followed by which the DNA polymerase comes here. it starts synthesizing the DNA from the 5 prime to 3 prime direction, and that is the complementary strand of the DNA. Okay, DNA ligase also comes into play in the replication process for joining the Okazaki fragments. That we'll also see in the future questions. Okay, RNA polymerase do not help in the DNA replication at all. Okay, so any doubts in this question? Okay, so let's move on to the next question. If two molecules of DNA double helix undergoes three rounds of DNA replication, how many molecules of DNA are created which do not have any original strand of DNA? So what can be the correct answer? Okay, okay, well, okay, okay. So the correct answer is option number two, that is 12. So let's see the explanation first. So as you know, the DNA replication follows the semi-conservative nature of replication. That means if there are two strands, then this is the old strands. Then the new strand that would be formed, that would have one old, one new, and the other one will be one new and one old. Okay, so this is called as a semi-conservative nature of replication, in which one old strand help in the formation of a new one, like this, and likewise. Okay, now if we consider this the question, so we have two molecules of the DNA double helix. For the first round of replication, we would have this kind of scenario. Okay, now for the second round of replication, we would have this one, this one, this one. This is from the first set. Now secondly, from the second set also. Okay, so this is from the second set. Now after the third round of replication, we would have, here would we have one. Okay, so this would be this one, three, four, five, six, seven, eight, nine, 10, 11, 12. Okay, so here we have 12 molecules that will be formed that would do not have any original strand. Okay. Now to calculate this we have a formula. Okay. So as we know, the replication follows the 2 to the power n uh, cycle. Okay, so we have that when there are number of cycles, so it the number of DNA strands would follow would be 2 to the power n. Okay, wait a minute. Yes, so this is for one DNA molecule that I am talking about. So, for example, if there are three cycles of the DNA replication, then eight molecules will be formed. Okay, so one, 2, 3, 4, 5, 6, 7, 8. Okay, now if we have to calculate the ones that do not have any old DNA strand, that would be okay. So this would yield us the number that six DNA molecules would not have any old strand in it, it would have only the new strands. Fine. Because we have two DNA molecules here, so this the total number would be 6 plus 6, that is 12. So is the explanation fine for this question? Okay. So let's move on to the next question. During the DNA replication, so what of the options are correct? Okay. Any more answers? Mhm. Okay. Okay. Any more answers? Okay. So the correct answer is option number 4, that is both A and C. And so as we can see here in the diagram also, once this DNA is actually being separated, the both strands are separated, the strand are need to be stabilized. Okay, otherwise it would denature in a short time. So the single strands are basically stabilized by the single stranded binding proteins. Okay, so that are called as SSBPs. So these SSBPs are helpful in stabilizing the DNA single stranded molecules during replication. Okay, now talking about the replication forks, so as we can see here, there are two replication forks that are being made in the DNA strand so that the DNA replication is bidirectional in nature. Okay, so there are two replication forks, this one and this one, that is actually being taken care for the replication to complete, and this is called as the bidirectional nature of the replication. Okay, so this is the uh, bidirectional nature of the DNA replication. Now, so the correct answer is option 4, that is both A and C are correct. Okay, so any doubts in this question? So if not, then we'll move on to the next question. Which feature of the DNA structure allows it to serve as a template for its own replication? So what can be the correct answer? Okay. 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 So the correct answer is option number 2, that is the complementary base pairing between the two strands. So what happens is, as you know, that whenever the DNA replication happens, so how would we know that what would be the base that comes to the other base? Because we know that in place of A on the opposite strand it would be T. On place of G, it on the opposite strand it would be C. Okay, and vice versa. So we know that the sequence of one strand would yield the sequence of the other strand but in the anti-parallel fashion. Okay, so that is why the complementary nature of the double helix will actually yield us the that actually tells us that the term template is being used to find to form the products in the replication of the DNA. Okay, so that is why the second option is correct. So is the explanation fine? Okay, so let's move on to the next question. Does DNA dependent DNA polymerase make mistakes while replicating the DNA? Okay, any more answers? Okay, so the correct answer is option A, that is first. So the DNA dependent DLF polymerase actually make mistakes, and that is one mistake per 100 to 1000 nucleotides. So these mistakes only help us in evolution process. Okay, so this is the first point. Secondly, it also leads to various diseases, the inborn diseases of the people. Okay, so these mistakes, because of the DNA dependent DNA polymerase actually being involved in various diseases that are genetic in nature, and these also help in evolution, that actually help in like transforming from one particular species to the other. Okay, so the DNA polymerase is actually not 100% proper or 100% sure that it will not actually cause any error. It also causes an error. Okay, so let's move on to the next question. Which of the following statements about DNA replication is not true? So, what can be the correct answer? Okay, so the four. Okay. So let's see the options. So Okazaki fragments are found on the lagging strands. Yes. So whenever the DNA replication is taking place, one of the strands will be continuous and the other one would be a discontinuous one. So this one that has a discontinuous type of replication have the Okazaki fragments. These are, after the replication is complete, these fragments are joined by a DNA ligase enzyme and to form a continuous DNA strand. Okay, so yes, this is correct. Then DNA polymerase replicates DNA from the 5 prime to 3 prime direction only. Yes, this is also correct. That's why the strand that is opposite to that, that is 3 prime to 5 prime strand, is actually forming the Okazaki fragments due to this reason only. Then DNA helicase unwinds and separates the two DNA strands. Yes, this is also correct. Then the DNA replication is continuous on both leading and the lagging strands. No, this is the false one. So the correct answer is option 4, that it is false. And here is the explanation for the same. So the DNA strand that is from the 3 prime to the 5 prime direction has a continuous DNA replication. Okay, that occurs between the 5 prime to 3 prime direction. The discontinuous replication happens in the opposite strand. Okay, and here it is the formation of the Okazaki fragments that take place, and these Okazaki fragments are later joined by the DNA ligase enzyme. And here, where the DNA helicase is actually being present, it actually helps in unwinding the DNA by breaking the hydrogen bonds, so that the DNA polymerase can actually sit here and start forming the complementary strands of the DNA. Okay, so any doubts in this question? Okay, so let's move on to the next question. Which enzyme joins the Okazaki fragments on the lagging strand? Okay, yes, this is correct. So the option correct is option number 4. So DNA dependent DNA polymerase helps in synthesizing the DNA. Helicase helps in unwinding the DNA. Primase helps in formation of the primer, because the DNA polymerase cannot initiate the polymerization process. So it requires a raw material that we call it as a primer. Okay, so that primer is generally RNA based and it helps in starting off the replication process. Okay, and DNA ligase helps in the joining of the Okazaki fragments to form the continuous strand. Okay, so any doubts in this question? Okay, so this was the last question for today. So if you guys have any doubt in the whole week or in the presentation, you can ask. Someone asked the doubt about the oh, central dogma. Okay, so what happens is, uh, yes, so I'll explain here only. So whenever, for this is a DNA strand. So whenever a DNA polymerase actually sits here, it will not be able to initiate the replication process. Okay, so for initiation, it requires a raw material that is actually being synthesized beforehand for that for the DNA replication DNA polymerase to actually add the nucleotide bases here. Okay, so there is an enzyme called as primase that actually synthesizes few nucleotides first, that are 8 to 10 nucleotides, and here the DNA polymerase would get a 3 prime OH group free here. Okay, now it is easy for the DNA polymerase to actually adds up the nucleotide here. Sorry. Okay, so is the explanation fine with that? Just 1 minute. That can be both assignment or problem solving based, or they would ask some different question but from the same pattern only. Those questions will be taught in the lectures. Okay, so that is not an issue. Okay, so someone asked about the central dogma. I'll go to that question first. Okay. So central dogma basically helps us to like understand the flow of information that takes place the from in the body. Okay. So what kind of information? So as we know, there is a source from which different phenotypic response that we see in our body. So that source is basically a gene or the DNA. So DNA is called as a like storage of the genetic information. So if we see DNA, it is basically helps in storage of the information. Storage of which information? The genetic information. Okay. So now what happened previously was this scenario was unknown, except that what molecule is actually being involved in the storage of information and which molecule is actually being involved in the movement of that information that leads to the phenotypic response of the body. So now Francis Crick, Sir Francis Crick, uh, okay, option 3, sorry, this one. So what do you didn't understand in this question? So, as I was saying, that DNA stores the information, and then following this, it transfers the information in the form of RNA. So RNA being an unstable molecule. Okay. So RNA is an unstable molecule that actually stores the information for some time only. Okay. Now that because it's unstable, it is a good molecule that actually help in transferring the information from one of the point to the other. Okay. So that it can quickly transfer the information from the storage unit to the expression unit basically. So now when the information actually transferred from DNA to RNA, this particular point is the deciding factor where it will be actually expressed in the form of a protein to show the phenotypic response or not. Okay. Now when the information is being transferred from RNA to protein, that means now the body has to show some phenotypic response. So this is actually the transfer of information from the storage unit to how it leads to the phenotypic response of the body. This is the main agenda of this particular central topic. Okay, so now it's clear. Okay. Uh, Replication in prokaryotes and eukaryotes. Which question can you please tell me? Oh, or you are asking the difference between the replication in prokaryotes and eukaryotes. Okay, so I think that will be discussed in the next lecture also. So we'll discuss that particular area in the next lecture because replication is not yet complete. So we'll discuss that whole thing in the next lecture also. Okay. So any more doubts? Okay. If not, then we'll close the session for today. So thank you guys for joining in. Let's see you next week. Thank you guys.